Evaluation of 2 genetic animal models in behavioral tests of anxiety and depression. Anxiety and depression-related disorders often appear associated and may be affected by common genetic factors. The inbred rat strains Lewis and spontaneously hypertensive rats and the outbred rat lines Florpa H and L, which were selectively bred for high and low locomotion in the central area of the open field test, respectively, have been proposed as experimental tools to study anxiety. The main goal of the present study was to characterize the behavior of these animals in 2 models of anxiety, the elevated plus maze and the open field, in 2 models of depression, the forced swim test and the tail suspension test, and in their home cages. Emotionality-related differences between Lewis and SHR rats and between Florpa H and L rats were found in the elevated plus maze, the open field and forced swim test. Those lines showing low anxiety-like profiles in the elevated plus maze and an open field, SHR and Florpa H, also showed low immobility in the forced swim test. The tail suspension failed to unveil any line differences. Factor analysis involving all tests revealed 3 independent factors, with one of them associating anxiety-related measures from the open field and elevated plus maze to immobility in the forced swim test. When observed in their home cages, Lewis and SHR rats showed no differences in general activity, but when acutely treated with imipramine, 15 mg/kg, only Lewis rats were sensitive to its antidepressant effects. These results suggest the existence of a genetic link between 2 tests used in the screening of anxiolytic drugs and 1 test of antidepressant activity. Moreover, the Lewis and SHR rat strains were shown to be an interesting model to study the comorbidity between anxiety and depression-related disorders.